So this is a frightening topic because it's moving so fast.
This should not be frightening. This is so there's CRISPR technology.
What, viruses
CRISPR,
attacking a cell is not frightening?
This
I'm
is the
frightened.
super cool part about CRISPR. CRISPR is basically an elaborate molecular strategy to recognize DNA from an invading virus, and the end result, a sliced apart DNA, and with the DNA cut in half essentially, this virus can no longer replicate its genome, and the infection fails, and the bacteria win. To cut DNA in a human cell, it turns out that cutting DNA acts as a trigger for various kinds of repair that allow you to end up introducing permanent changes to the genome.
I was particularly interested in the embryonic aspect and editing before birth.
My lab and my colleagues developed a system where we can generate artificial human embryos. It's a human embryo that's made of human embryonic stem cells. But what I use CRISPR-Cas9 in this case is to figure out where the lineages come from. How does a cell inside of an embryo decide if she's going to be blood, bone, muscle, or brain, or skin?
How does Big Pharma respond to something like this?
What CRISPR did is really cut decades of work into, or I would say years of work into days. When you wanted to understand the function of a new gene, or it used to be a PhD thesis, right? And then it became maybe something that takes a year. And suddenly, within 3 weeks, you covered 20,000 genes, and you knew exactly what was going on. This is revolutionizing our ability to identify new biological mechanisms and disease mechanisms.
He claims that there are twin girls that have been born who have had their genome edited. It is absolutely the first time that anyone acknowledges in history that this has happened. There's no question that the international reaction was one of condemnation and shock. He was editing specifically because the father had HIV, and he was editing so that the embryos had the CCR5 gene, which is people who are naturally immune. And so he essentially gives them this immunity. So his claim is that there are no deletions, zero complex mutations, zero ramifications of it. Do you believe this?
So I think a lot of news stories, they say CRISPR is this highly precise, amazing gene editing tool. It is not actually that easy in practice. There are things that have become incredibly easy, like if you want to make a knockout mutation and you want to just get rid of a gene, CRISPR is fantastic for that. But if you want to talk about making a therapeutically safe edit or mutation, it's not ready for prime time.
If you were to do this experiment in any other animal model systems, what you will very quickly see is when you inject the tools for CRISPR-Cas9 in an embryo, you don't get a global effect in all the cells. You get this thing called mosaicism, where some of the cells have the CRISPR-Cas9 edition, and then the other ones don't. And as far as we can tell so far, seems to be a random process. It's impossible to think, again, one gene, one function. If you think of the keyboard and the music, changing a key on the piano will have consequences that would affect all musics. So one gene that is a function within the first week of development will have a completely different function once the child is born, and then even more different functions after puberty, let's say. So the idea that we can fix in the case of CCR5, that somehow this is going to fix AIDS, completely ignores that CCR5 has functions during early development that are necessary.
How do you deal with the rogue experimenters when it's so cheap and easy?
It seems to me that there was the appropriate reaction to this experiment worldwide. It was not just the US that condemned. It's definitely a very disruptive discovery, but there will be some evolution. Really, what justifies intervention, right? I mean, is it the desire to have a more beautiful baby? Is it really formal disease? Where does a condition or a disease start and quality of life, and
What about the idea of designing our successors? What if we want to make astronauts with a different bone density who require less food, air, and water? We want to design human beings for specific tasks.
The early study of the human embryo allowed us for the first time to discover organs that we never imagined existed. And it's, at least to me, amazing to think of how many of the organs in our anatomy are transient organs that you never see because it develops in the womb and it disappears before birth. We're beautiful, as I said. After a month, we have a tail. We have a gorgeous tail.
Yeah.
We have
Gills.
gills.
Fascinating.
We actually breathe in water. These are natural attributes. These are not CRISPR-Cas9 mediated changes. These are the information is within our DNA as we speak right now. It's not impossible to imagine, and in fact, I like to think that it's absolutely possible to reawaken that information.
100,000 years, 1 million years. I mean, if humans are able to stick around that long, I think the days when it's just natural sexual reproduction that leads to the next generation, those are over. I mean, it's 2% of births in the U.S. today are by IVF, 2%. But the technology is getting better and better, and soon you're going to be able to say, here's a panel of genes and gene variants that the two parents can contribute. So even without CRISPR, I think we're going to see a changing world in that regard. Disability rights is a major stakeholder in this issue, and I think that's one of the more ethical or philosophical concerns, that the idea of being able to select the genetics of your child will now promote the idea that there are certain children with genetics that are better than others.
I think autism is a wonder. It's an enigma, because it is, to classify it as a disease is very superficial. I mean, I think it's something we don't understand. There is a lot that we don't know about biology and the human condition that will unfold in the coming decades, thanks to advances in science like CRISPR-Cas9.
I was once in a similar conversation with somebody who I won't identify, who's a brilliant geneticist, and somebody from the audience said, well, wouldn't you eliminate autism if you could? And he said, I'm autistic. Okay, and that was like a moment. So what do you mean genetically superior embryo? Do we know what that means? Do we look at that embryo and say, that embryo has autism? Okay, and there goes one of the greatest minds of our time.
There are mutations that are lethal. For example, if you consider Huntington's disease, this is a single gene mutation with 100% penetrance. That means if you have the mutation,
you get the disease.
You're out of luck. I think in that particular case, we can agree that if we can correct something that otherwise would not allow the development to continue or the children to not die early on, then you can ask yourself, well, if I have a technology that can save life, and if I don't use that technology, is that really ethical?